Hi, everyone. I'm Liang from GemPharmatech. I'm glad here to talk about the selection SLE mice model for preclinical efficacy study when considering drug mechanism of action. So first, I will give a brief introduction about SLE. SLE is a serious chronic autoimmune disorder with autoantibody production and immune complex deposition, followed by damage to target tissues. It's a quite complex heterogeneous disease, potentially involve many organs. In SLE, the eye, skin, kidney, toes, joint, the cardiovascular system, and the brain could be also involved. Each patient demonstrating a distinct pattern of disease. SLE is caused by an autoimmune reaction that involves innate and adaptive immune systems. Also, the pathogenic mechanism of SLE is quite complicated. The type 1 infron and autoantibodies, for nucleic acid and nucleic acid bind proteins have been considered as the two fundamental contributors to the disease. When innate and adaptive immune systems are engaged and collaborate in the autoimmune response, clinical SLE could develop. The genetic variants are very important risks and triggers for SLE development. Insufficient availability and function of complement components gene, such like C1Q, C2, and C4, are associated with SLE development. The dysfunction of DNAs that degrades DNA, like TRIX1, can also cause a rare lupus-like disorder in patients. The environmental triggers, such as the UV light and also the drugs that mediate DNA damages, may generate the stimulatory self-nucleic acid. The self-nucleic acid might work as the autoantigens to trigger immune response. Beyond these factors, the potential microbiome triggers like EBV could also directly promote type 1 interferon release from PBC. The self-nucleic acid is main drivers for SLE development. It might be generated by DNA breaks, the oxidized mitochondrial DNA, the necrotic and apoptotic cells. The self-nucleic acid could interact with autoantibodies to perform immunocomplex. The immunocomplex could be delivered to TLR7 and TL and toll-like receptor 8 through FC receptors and activation those signaling pathways, leading abundant release of type 1 interferon from PDC and also the TNF from monocytes. The type 1 interferon could promote the presentation of antigens through DC cells and leading to the activation of T cells. B cells are driven to promote abundant autoantibodies through interaction with T cells and also the cytokines released by DC cells. The long-lived plasma cells has been considered to the main source for the generation of autoantibodies. The autoantibodies, especially those autoantibodies complex with the nucleic acid, could form the immunocomplex. The immunocomplex are the essential mediators that induce tissue inflammation and also the organ damage. The immunocomplex could be deposited in the kidney to develop the lupus nephritis and also cause damage to the other organs. So far, there is no diagnostic criteria available. The diagnosis is based on clinical manifestations and the laboratory testing, including measurements of autoantibody titers. The high level of autoantibody, the level of autoantibody is also the parameters that could indicate the disease activity of the preclinical animals. The current guidelines recommend guideline recommend to treat lupus, including using the non-steroidal anti-inflammation drugs, anti-malarial drugs, glucocorticoids, and also the immunosuppressive therapies. The biologicals and the targeting drugs are also very important for the treatment of SLE. The current drug pipelines working on the inhibition of type 1 interferon systems and also working on the inhibition of components of the adaptive immune systems. For inhibition of the type 1 interferon systems, the drugs could be designed to blocking the function of PDC through anti-PDCA2 antibodies. The PDCA2 is markers of PDC. The drugs could also be designed to blocking the toll-like receptor 7 signaling pathway, reduce the release of type 1 interferon. And also drugs could be designed to blocking the signaling pathway of type 1 interferon receptors and also the downstream JAK signaling pathways. For inhibition of the components of adaptive immune systems, the strategy could be to blocking the function of the T follicle helper cells through anti-CD40L, anti-ICOS. BCLs is also the very important target for treatment of SLE. The bimumab is the BAFF antibodies that have been proved for prevent the survival of autoantibody survival, autoreactive BCL survival. The other strategy targeting for BCL, including blocking the BCR signaling pathways through BTK inhibitors, and also removal of BCLs through CD19 CAR T cells, or the CD22 antibodies rituximab to remove BCLs. The long-lived plasma BCLs has been considered to be the major source for autoantibody generations. So the drugs that targeting long-lived B cells through anti-CD38 and BCMA are also the promising therapeutic for treatment of SLE. Here is a summary of the current drug pipelines. The drugs that targeting for B cell activity, BTK pathways, the JAK pathways, the type interferon, and also TLR7, and also TLR signaling pathway are still getting lots of interest by the marketing. So here is the SLE mice model we have generated in GemPharmatech. So far, we have generated 4 different SLE models. Each model has distinct therapeutic mechanism to develop SLE. With analysis of the drug mechanism of action, we could choose the drugs. We could match the drug with the suitable animal model with the compatible genetic mechanism to facilitate the drug efficacy study. For example, if the drug is targeting for the type 1 interferon or the drug is the JAK inhibitors, we will recommend the B6 TRIX1-KO mice for the drug efficacy study, since the TRIX1-KO mice is featured when the C-gas sting and type 1 interferon activation. If the drug is the BAFF antibodies or the PDK inhibitors, we will highly recommend to use the BAFF humanized mice model to evaluate such drug efficacy, since this B6 humanized BAFF animals is characterized with the B cell activation. The pristane-induced SLE model is featured with activation of the innate and adaptive immune systems. So if the drug is the targeting for the JAK inhibitors or targeting for the Toll-like 7 and 8 antagonist, we will recommend the pristane-induced SLE model for evaluation such drugs. The induced-HSC-NCG-M mice model has the humanized immune systems, and in this model, the PDC subpopulation could be activated. So if the drug is targeting for the BDCA2 or targeting for the type 1 interferon receptor, we will recommend to use the induced HSC-NCG-M mice to evaluate such drug efficacy. Here I will give the details of our SLE mirror models manifestations, and also give some case for the drug efficacy study. The first SLE mirror model is the humanized BAF mice. The BAF is the B-cell activating factors. BAF is a B-cell survival factor when supports autoreactive B-cells and prevents their depletion. We generate the humanized mice model with random insertion of the human BAF coding sequence into the mice genome. The humanized BAF mice is featured with a high expression of human BAF. We have generated two different lines of the BAF TG mice. And in this BAF TG mice, we first examine the level of the antibodies in the serum. The first three group as a female group, and the other three group as male group. The black one is the B6 control mice. This one is a BAF TG line 4 mice. This is the line 35 TG mice. Similarly, is B6 control mice, line 4 TG mice, line 35 TG mice. As shown in the figures, the serum IgM levels are increased in the BAF TG mice compared with the B6 control mice at 6 weeks, 26 weeks, at 6 weeks, 40 weeks, 26 weeks, and also 34 weeks. The serum level of IgM were increased continually with transgenic mice aged. And also we observed similar results of IgE level in serum. The IgE level in the TG mice were also increased at 6 weeks, 14 weeks, 22 weeks, and also 34 weeks. The IgE level in the serum will increase gradually when transgenic mice aged. Similar results was also observed in the IgG level in the serum. For the serum anti-dsDNA level, we also found that the TG mice showed increased dsDNA level in the serum compared with the control mice at the 6 weeks, at 8 weeks, 16 weeks, 24 weeks, and also 34 weeks. The serum anti-dsDNA levels keep at the high level during the whole study period. And more importantly, these humanized BAF mice develop lupus nephritis at around 25 weeks. The pathogenic HE study showed that the B6 BAF mice could show the glomerular enlargement, and also it displayed the immunocomplex deposition, and also it forms it also displays the crescent formation. Those pathological change has also been observed in the patients. So the B6 humanized BAF mice could develop the lupus nephritis that resembles the phenotype of patients. We also detect the complement components C3 deposition in the mice, and we found increased level of C3 deposition in the B6 humanized BAF mice. The statistic work showing the increased lupus nephritis activity index in the TG mice compared with the control mice, and also the lupus chronic index also increased in the TG mice compared with the B6 control mice. In the applied drug efficacy study, we use BAF antibody and BTK inhibitor to treat the BAF TG mice. The first four group as the female mice, and the other four group as male mice. The first one is B6 mice, the BAF TG mice. The BAF TG mice treat with BAF antibody. The BAF TG mice treat with BTK inhibitors. As shown in the data, the BAF antibody treatment could reduce the level of anti-dsDNA, IgA level, IgG level, and also IgM levels in the female or male B6 BAF mice. And the BTK inhibitors could reduce the level of IgG level and also the IgM levels in B6 humanized BAF mice. So summary here, the B6 humanized BAF mice is featured with the increased B cell population and also activation of B cells. So this model could be very useful for evaluations of drugs that target for B cells for treatment of SLE, especially the drugs that target for BAF or the drugs that target for PTK. The second mice, the second SLE mice is B6 TRIX1-KO mice. TRIX1 is a DNA nuclear exon nuclease. It plays important functions in the genomic DNA degradation. Dysfunction of TRIX1 may activate immune response by self-DNA. We generate the TRIX1-KO mice with remove the exon of these genes. We confirm the knockout of the protein by Western blot results. So we detect the anti-DNA level in this model. The first three mice are the female mice. The other three group as male mice. The first one is the B6 control mice. This is the hetero-TRIX-KO mice. The third column is the homo-TRIX-KO mice. As shown in the study here, the TRIX1 homo-KO mice showed increased serum anti-dsDNA levels at 7 weeks, 9 weeks, 13 weeks, and 21 weeks. And we also perform the blood biochemistry studies in these animals. We detect ALT, AST, and LDH for the study. Those three parameters could be the indicator of the function of heart. So in these results, we found the significant increase of ALT, AST, and LDH in the TRIX1 homo knockout mice that indicate the dysfunction of the heart in this model. And also during the study period, we found the increase of the blood biochemistry parameters lasting around 40 weeks. In the pathological analysis, we found the inflammation cell infiltration in kidney, heart, liver, and spleen at around 8 weeks. So that indicated the inflammation of multiple organs in this model. And we also performed the pathological analysis at around 32 weeks. At 32 weeks, In the 32 weeks pathological analysis, the heart also showing the cardiomyocyte necrosis and inflammatory cell infiltration in the skin. Then we try to figure out the pathologic mechanism in these TRIX1-KO mice. And with the detection the expression of the interferon stimulatory DNA response genes, we found all the tested interferon stimulated DNA genes are all increased in the TRIX1-KO homo-KO mice. That indicates the type 1 interferon signaling pathway has been an activation in these TRIX1-KO mice. The study was performed at 32 weeks in the spleen tissue. The similar results were also observed in the heart tissue at 32 weeks. In the applied drug efficacy study, we use the anti-malarial drugs and also the JAK inhibitors to treat the TRIX1-KO mice. The first 4 group are the female mice, the other 4 group are male mice. The first is B6 control mice, the TRIX1-KO mice. The TRIX1-KO mice with treatment of anti-malarial drugs, the TRIX1-KO mice treatment with JAK inhibitors. So as shown here, the ALT, AST, and LDH were reduced with JAK inhibitor treatment in the TRIX1-KO male mice. That indicates the JAK inhibitors could rescue the heart dysfunction in this model. The anti-malarial drugs also showed therapeutic effect in the TRIX1-KO model in both female and male group. So summary here, the TRIX1-KO mice is featured with activation of the type 1 interferon signaling pathway, with activation of the C-gas STING signaling pathway. So the drugs that targeting for the C-gas STING signaling or the type 1 interferon could be used this model to evaluate the drug efficacy. The third model is a pristane-induced SLE model. Pristane is a well-used chemical to induce SLE in mice. In this study, we performed the experiment in 3 different inbreed strains, C57, PBC, DBA. We induction the model for 9 weeks and the detection of level of dsDNA at the groups of the mice with drug treatment. During the study experiment, we detect the protein urine, anti-dsDNA level, anti-nucleosome, and also the level of anti-Smith protein. At the termination, we collect the spleen and kidney for the pathological study. In this model, we found the PBC and DBA mice are more sensitive to the treatment of pristane. As shown here, in around 9 weeks, the pristane could induce increased level of dsDNA in the PBC mice and also the DBA mice, but not in the B6 mice. The increased dsDNA level were also observed in 13 weeks, 17 weeks, and also 21 weeks in the PBC mice. So we will recommend PBC mice to perform the pristane-induced SLE model. Also, we detect the anti-nucleosome antibody levels in those 3 models. And for these parameters, the DBA are still showing the sensitive to the pristane induction. For the anti-Smith protein levels, we found the increased anti-Smith protein levels in PBC mice and DBA mice after around 17 weeks induction, and also the increased level of anti-Smith proteins were observed in the 24th weeks. We then performed the pathological analysis of kidney at around 19 weeks. The pristane-induced group all showing the increased level of inflammatory cells infiltration. And then we detect the pathogenic mechanism in this model. We detect the interferon stimulatory DNA levels in this model, and we found the TNF-alpha and the CCL2, those two interferon stimulatory DNA all increased in the pristane-induced group in the three inbred strains. In the applied drug efficacy study, we use the TLR7/8 antagonist and the JAK inhibitor to treat the model. The first one is a vehicle control. The blue column is a pristane-induced model. This is a pristane-induced model with treatment of TLR7/8 antagonist. The last one is a pristane-induced model with treatment of JAK inhibitors. The anti-nucleosome levels was reduced with toll-like receptor 7/8 antagonist treatment and also the JAK inhibitor treatment. The protein in urine showed a decreased trend with treatment of the toll-like receptor 7/8 antagonist. So summary here, the pristane-induced SLE model is featured with the enhanced BCR and the TLR7 signalings. The model could also show an increased release of type 1 interferon. The model also showed increased activation of type 1 interferon. So this model could be used to evaluate the drugs that targeting for the toll-like receptor 7/8, the BTK inhibitors, and also the antibodies that targeting for the type 1 interferon receptors. As reported by the literature, the pristane could induce the proliferation of the membrane-like B cells. So the antibodies that target for B cells, like the antibody targeting for CD20, CD40, CD38, could also be evaluated in this model. The last model of SLE is induced-HSC-NCG-M model. First, we generate this model through transplantation of the HSC into the NCG-M mice. NCG-M mice is the immunodeficient mice. With transplantation of HSC, it could reconstitution the human immune systems. And then we administration the mice with toll-like receptor agonist. The toll-like receptor agonist could activate the PDC with release abundance of type 1 interferon. The type 1 interferon could promote the antigen presentation by DC cells, leading to the T cell activation and also the B cell differentiation. The mature B cell could, the autoreactive B cells could generate the autoantibodies. So this model could develop SLE. Here is just a brief introduction about the HSC-NCG mice. NCG mice is a severe immunodeficient mice with expression of the human cytokine like SCF, IL-3, and GM-CSF. This NCG-M mice could be very useful for reconstitution immune systems. When transplantation the HSC into these mice, these mice could develop several different immune cell populations, like the CD3 positive cell, CD19 positive cell, and more importantly, this model could develop functional DC cells. So in this HSC-NCG mice, after reconstitution of the human HSC, we administer the mice with the TLR pathway agonist. The administration of the TLR agonist could develop a SLE in this model. And we evaluate the phenotype of the mice and do the drug efficacy study. As shown in the figure here, this model could develop the CD T cell population and also B cell populations. And when the TLR agonist treatment, this model could also develop PDC cells, and this model showing increased level of the IgG in the serum. The induced HSC-NCG-M model displays rash on the face and back, and also it shows a hair loss on neck and back. The IgG1 immunocomplex deposition was also observed in kidney in the toll-like receptor agonist treatment group. And in the drug efficacy study, we use the BAFF antibodies and also the CD20 antibodies to do the drug efficacy study. As shown here, G2 and G3 groups are the BAFF treatment group and the CD20 antibody treatment group. As shown here, the BAFF antibody and the CD20 antibodies could all reduce the IgG level in the SLE model, and also as shown in the yellow and blue curve, the anti-dsDNA levels were also decreased after drug treatment. So here is a brief summary here. We have generated 4 different SLE animal models. Each animal model has distinct pathogenic mechanisms. Like the TRIX1 kill mice, it is featured with activation of C-gas stain type 1 interferon. The humanized BAFF mice is characterized with activation of B cells. The pristane-induced SLE model is showing activation of innate and adaptive immune systems. And the induced-HSC-NCG-M mice, it has the humanized immune systems, and it also has the activation of PDC. So with analysis of the drug mechanism of action, we could match the drug with the suitable animal models to facilitate the drug efficacy studies. If the drug is targeting for the type 1 interferon or the JAK inhibitors, we will recommend the TRIX1 kill mice. If the drug is specific for the BAFF or it is BTK inhibitors, we will highly recommend using the B6 humanized BAFF mice. If the drugs are targeting for the TR7 and 8, the pristane-induced mice model could be suitable for this drug evaluation. And more importantly, we can also use the humanized CD20 mice to induce SLE phenotype with the pristane. So these humanized CD20 mice could be used to evaluate the drugs that targeting for CD20. And also for the drugs that targeting for the BDCA2 or for the PDC cells, we will recommend to use the induced HSC-NCG-M mice. Last, I would like to introduce the platform for the autoimmune disease in GemPharmatech. Now we can provide the one-stop solution for autoimmune drug preclinical research. It including the preliminary screening, the model construction, phenotype validation, and also the drug efficacy evaluation. Besides the SLE model I have introduced, we also generate several different autoimmune disease models, like psoriasis model, atopic dermatitis model, the UC model, CD model, also the asthma models and the EAE models. For each disease, we have generated multiple different models with the distinct pathogenic mechanism. So this will be very helpful for the drug efficacy study. The advantage for our service is to do the drug mechanism of action analysis. We will help our clients to choose the suitable animal model based on the drug MoA to help get the good results for the drug efficacy study. We also have the QAP resource for the autoimmune disease pipeline. QAP is a knockout out project. In this project, we aim to generate around 22,000 kill of flux mice with knockout of all the protein coding sequence. The knockout mice could be the spontaneous autoimmune disease model. And also, we have generated the humanized genetic engineering mice model. As examined here, we can use the BBC humanized IL4R model to induction atopic dermatitis. In this humanized autoimmune disease model, we can evaluate the drugs that targeting for IL4R for the treatment of atopic dermatitis. And also for the BBC IL6 mice, we could develop arthritis in this model. So we can evaluate the drugs that targeting for IL6 for treatment of arthritis. The immunoreconstitution platform is also the very important resource in the autoimmune platform. In this immunoreconstitution platform, we could generate several different cell subpopulations. We can induction autoimmune disease in this humanized mice model. So this animal model could be very useful to evaluate the humanized antibody. Also, we have the microbiome platform. In this microbiome platform, we could evaluate the microbiome efficacy for treatment of autoimmune disease. So here is all the resource we got in the autoimmune platform. Now I'm ready to take the questions. If no more questions, I think maybe we can stop the webinar.